So today we are going to discuss about a further concept of mitochondria. So previously we discussed about some aspects such as how it is containing its structures, what kind of structure is present inside its inner mitochondrial membrane, as well as whether it is a uniparental or biparental inheritance, how is it a uniparental inheritance, and such things were discussed about its semi-autonomous structure and such stuff. Now we are going to discuss about its endosymbiotic nature. Scientists believe that according to the endosymbiotic theory, mitochondria are derived from prokaryotes. They believe, according to endosymbiotic theory, mitochondria were prokaryotes priorly, were prokaryotes. According to endosymbiotic theory, these are prokaryotes in actual. Mitochondria and chloroplast are and and chloroplast are prokaryotes in actual. How do they believe that? Because as we know that inside the structure of mitochondria there is double-stranded DNA which is circular, guanine, cytosine rich, which is same characteristic of prokaryotes. Secondly, it is having RNA of all three types which is possessed by prokaryotes as well. It is having ribosome 70s which is possessed by prokaryotes as well as it is having several enzymes such as RNA polymerase 1 enzyme which is only present in prokaryotes, and that is the only enzyme present in mitochondria and chloroplast as well. Fifthly, it is having DNA polymerase enzyme which is present in prokaryotes as well as it is able to self-replicate. It is able to produce its own protein, while thirdly it divides by binary fission, which are all the characteristics of prokaryotes. In this manner, they believe that mitochondria and chloroplast are prokaryotes in actual, because nearly every aspect of mitochondria and chloroplast resembles with that of the prokaryotes. So in general, prokaryotes are the most primitive living organisms on earth. These are the most primitive living organisms on earth, or the first living organisms which evolved on earth. These are the first living organism evolved on earth, or the most primitive living organisms on earth. Most primitive living organisms and the first organisms to evolve on earth, after which mitochondria and chloroplast were basically considered in such a manner that, for suppose this is mitochondria. Now this mitochondria previously was considered to be this mitochondria was previously considered to be aerobic bacteria. Aerobic bacteria. When it comes to the concept of prokaryotes as the first organisms to evolve, which prokaryote was the first organism to evolve? It was archaeobacteria. Now aerobic bacteria was the first organ. Aerobic bacteria was considered to be mitochondria. Now that was able to do aerobic respiration, which means it was able to respire in presence of oxygen. So in that case, um, this was considered to be aerobic bacteria. Now on the other hand side, there was this. For suppose this is chloroplast. Um, Chloroplast. Let me just draw the structure. For suppose this is chloroplast, and here within the structure of chloroplast, this was considered to be previously as photosynthetic bacteria. Why? Because it is containing the chemical synthesizers, which are in actual the pigments. These chemical synthesizers are pigments which absorb specific wavelengths of radiations of light, which is responsible for storing the energy required for the process of photosynthesis. So in that case, these were the organisms. On the other hand side, when it comes to The major concept of endosymbiotic theory was such that in the endosymbiotic theory, the scientists believed that this was the first eukaryotic cell. The early eukaryotic cell was in such a manner that this was the early eukaryotic cell, it was its nucleus. Now that early eukaryotic cell the engulfed the mitochondria and it engulfed the chloroplast. But as mitochondria aerobic bacteria was doing aerobic respiration, was respiring in the presence of oxygen, it instead of digesting the mitochondria and chloroplast, it provided it a permanent space within its cell, due to which the early eukaryotic cells adopted the aerobic bacteria and photosynthetic bacteria as their permanent organelles, because they were providing benefit to it. Mitochondria was providing aerobic respiration through which energy was produced, whereas chloroplast was providing food to it in the form of glucose. So in that case, both of these were considered to be beneficial for the early eukaryotic cells, aerobic and photosynthetic bacteria. And when these were allocated a personal or permanent place within the early eukaryotic cell, then the early eukaryotic cell was converted into the evolution of an eukaryotic cell, in which there was nucleus, mitochondria, and chloroplast. Now this eukaryotic cell was containing an organism which was aerobic bacteria and an organism known as photosynthetic bacteria within the eukaryotic cell, which was considered to be an endosymbiote, which means organism within organism, endosymbiote. Now, after that, as we discussed about the enzymes, there are three. There are majorly three RNA polymerase enzyme: RNA polymerase 1, 2, and 3, which are present in eukaryotic cells, but in um, the prokaryotic cells, there's only RNA polymerase 1. Now, in this case, in order to substantiate this experiment, they took the eukaryotic cell. Scientists took the eukaryotic cell and applied a chemical within the eukaryotic cell, dropped a chemical on the eukaryotic cell known as chlorophenicol. This is an antibiotic which is able to inhibit the protein synthesis of bacteria. It is an antibiotic which is able to inhibit the protein synthesis of bacteria. What does it mean? This means that when it was dropped over the eukaryotic cell, the eukaryotic cell's protein synthesis did not inhibit, but the protein synthesis of the mitochondria and chloroplast was isolated and stopped, due to which it was a true evidence that mitochondria and chloroplast possess the characteristics same as that of the prokaryotes. So in that case, it was found that mitochondria and chloroplast are derived from, not derived from, but these are in actual the prokaryotes. Mitochondria and chloroplast are in actual the prokaryotes. I hope that sounds clear. After which we are having the enzymes RNA polymerase 1. So DNA makes RNA, RNA makes protein. When DNA makes RNA, it's known as transcription, and that requires the enzyme known as RNA polymerase. And that RNA polymerase can also be written as DNA dependent RNA polymerase, because DNA is required to form RNA by the enzyme DNA dependent RNA polymerase. So it can also be written as transcriptase, because it is undergoing the process, or it is causing the process of transcription. So it can be written as transcriptase. Now RNA, when it is converted into protein, it is known as translation. But here we are going to only discuss about RNA. So here RNA, for suppose RNA is made using the DNA dependent RNA polymerase. Now sometimes RNA converts back into the DNA. RNA converts back into the DNA through the process which is known as reverse transcription. So this is the process which we call as reverse transcription. And in the process of reverse transcription, there is an enzyme working known as DNA polymerase. DNA polymerase is an enzyme which DNA polymerase is an enzyme which converts the RNA into DNA by producing the deoxyribonucleotides, while RNA polymerase or DNA dependent RNA polymerase converts DNA into RNA by forming ribonucleotides. Or in other words, we can call that as ribonucleotide monophosphate. So DNA is converted into RNA by formation of deox by the formation of ribonucleotide monophosphate. DNA into RNA by formation of ribonucleotide monophosphate, which includes ATP adenosine triphosphate, GTP guanosine triphosphate, UTP uridine triphosphate instead of thymidine triphosphate. After which there is CTP cytosine triphosphate. After which these are the nitrogenous bases. These are the nucleotides involved in the ribonucleotide monophosphate. After which, when we convert RNA into DNA, this process is known as RNA reverse transcription, in which the enzyme DNA polymerase works. But this time, DNA polymerase basically forms the deoxyribonucleotides. In that case, the DNA polymerase forms deoxyribonucleotides, after which this deoxyribonucleotide is formed by DNA polymerase, which can also be written as RNA dependent DNA polymerase. This can also be written as RNA dependent DNA polymerase, RNA dependent DNA polymerase, or we can also write it as reverse transcriptase because it undergoes reverse transcription. By the way, reverse transcription occurs in HIV and retrovirus. And yeah, so this is a brief concept about the this is a brief concept about endosymbiotic theory. After which yeah, there's another thing that DNA is considered to be genotype, and after genotype there are two gaps between the genotype, RNA and protein, and then protein basically causes the character expression, which is the phenotype, the physical expression of the character, and genetic expression of the character is genotype, which is in the DNA. So hopefully that's all about this video, and we are going to move and meet again in another one.